Hello guys, it's Adam the Science Student here, and today we're going to be going over the reproductive system and the different hormones that will lead you to becoming a phenotypically male or hermaphrodite in some cases, or phenotypically female. But genetically, you are either XX or XY in these circumstances. So to start things off, we have our two end products, or not products, but end sites. So we're going to end at the female internal genitalia for women, which is their ovaries and whatnot. It's internal because they also have their exterior breast, but we're talking about their internal genitalia. And then for the male, it's going to be the penis prostate, and that's going to be where the hormones are going to end on that spectrum. Start things off, we're at the genital edge. This is like our first 2 weeks of being an embryo, so we're not yet established, we're not even differentiated yet. Our gonads in either male or female genotypically have yet dropped. So you could be an XX female, but you're undifferentiated. You don't have gonads that have dropped yet. And our important hormone to make us become differentiated is WT1. So WT1 is our big time hormone factor that plays a role in differentiation. So it's for men, your genitalia will drop to your testes. For women, your genitalia will drop to your ovaries. So then we have, like I just said, our bipotential gonads. So we're gonna go here, make sure we have the firm. Okay, so let's do the men's hormones first and men's cycle first. So there's two important hormones for men. It's going to be the SRY gene and the SOX9 gene. These are both extremely important. These will make up the Wolffian duct, and that we're going to talk about in a little bit. But these are extremely important to the differentiation of becoming a male. If you lack the SOX9 gene, you will be genotypically a male, you'll be an XY. However, you'll be phenotypically female. This is seen with hermaphrodites, and this is a real cause of concern. If a female is born and has the SOX9 gene on her, she will be XX genotypically, but she will express male phenotypic traits. So she would be not able to have birth. She would have not the Mullerian duct, which all females need to reproduce and all females need for their correct life cycle. So these are two very important genes. And oftentimes a male born without a SOX9, they will look so phenotypically female that families or doctors won't be sure until maturation up to puberty hits, and they will see that the genotypically male is unable to have a period, therefore missing its SOX9 or SRY gene. So with these, we're going to create our testes. So these are testicles. This is our main dudes for our sperm creation and our way of life. Testicles are going to go out and create two different types of cells. They're going to create the Serotoli cells. Then we'll have our Leydig cells. Our Leydig cells. And Leydig cells are important for testosterone creation, which we all know how important testosterone is for men. So Leydig cells will create testosterone. And these SF1, they're both transcription factors, so they just help these cells mature. So it's not a big portion. You don't have to memorize that hormone or that transcription factor, but it is what it is. So Leydig cells, as I just said, make some testosterone. And then testosterone goes to DHT, which I'll get to in just a sec. Serotoli cells will go to AMH, which is an anti-Mullerian hormone, so it makes sure that you're not creating a Mullerian duct. Testosterone goes straight into the Wolffian duct. This is what makes us a male completely. This is our Wolffian duct. So males have the Wolffian duct. Females we'll get into in a bit, but they are gonna have the Mullerian duct. And then, yeah, these all go straight to penis prostate. So DHT is good because it's the androgen sites for or receptors for testosterone. So it's where the testosterone will go, and that's where our body can utilize it. So it's very important. DHT's it's seen in male pattern baldness. So yeah, kind of cool. All right, so finished up with the male hormones. That's how we develop into the penis prostate. So just a quick review. We get the Wolffian duct, extremely important. We have a couple important things along the way. WT1 creates differentiation for both. SRY, SOX9 are very important for male differentiation, so males need this to create. These are directly involved with the creation of the Wolffian duct. So these are important for the Wolffian duct. The testes are developed. You create from the testes cells. From the testes, you create your Sertoli cells and your Leydig cells. Sertoli cells create your anti-Mullerian hormone, and Leydig cells create your testosterone. Testosterone is read and received at the DHT, which is going to receive it and use it for the penis prostate. So, women will have DAX1 to create their ovaries. And the ovaries, similar to the testes, will create two different cell types. So that's kind of good. And these are the theca and granulosa, or also called follicle cells. Theca cells. Cool. So follicle cells are made to produce your androgen or produce your estrogen. Theca cells is to read it, to understand it, and to break down the estrogen. So these are both going to create. So we're in a room. Follicle cells. Or your follicle, my bad. It'll create your follicle, which is found in the ovary. The follicle will read, digest, make some estrogen. As soon as you see the estrogen, as soon as you saw the testosterone, it's our Wolffian duct. This is going to be our Mullerian duct. Mullerian duct. And then it goes from estrogen to the female internal genitalia. And it's once again it's internal because that is her ovaries, it's not her breast. But these are largely largely controlled by the hormones at play. And these genes are exactly what makes us who we are. So DAX1 is important for women development into the ovaries. The Mullerian duct will not be formed if the SOX9 happens to be a mutation, and then let's say there was a SOX9 gene here, this would not create. So it's very important to have that down or have that understood. And just similarly, if the SOX9 is missing here on the genotypically male, if a SOX9 is missing, Wolffian duct is gone. If SRY is missing, Wolffian duct is gone. So you need both of these for a male. For a female, you don't want to have the SOX9 or SRY gene in you either. This is genetically this is a bit rare, so it doesn't happen often, but it does occur. It creates your hermaphrodites, and there's also a disease called campylemic dysplasia, which is reduced if you don't have a SRY gene. So just things to keep in notice, but this is it. I know I kind of scratched it up a bit now, but I could fix this. But yeah, so we have our SRY and SOX9, produces our testes. We want to create the Wolffian duct. Create Leydig cells, Sertoli cells. This is an anti-Mullerian duct hormone. So Sertoli cells, which are the nourishment of our sperm. It also creates the AMH hormone. Leydig cells create our testosterone. Testosterone makes DHT. Too much DHT leads to male pattern baldness. And then it goes to penis prostate. Mullerian duct. So our female duct may establish with estrogen. That's what makes you female, male, male, female. So yeah, if you guys have any questions, just send me an email. I know this can be a little confusing, but your main hormones are listed here. WT1 differentiates, so that's important. DAX1, female, creates ovaries. SRY SOX9, male. You create your granulosa or follicle cells, or theca cells for females, your Sertoli cells and Leydig cells for males. AMH is just an anti-Mullerian duct hormone. DHT leads to male pattern baldness. Our main factors here, we create our estrogen in females and testosterone in males. Both of them are extremely important in our anatomy. But thank you guys for watching. It's Adam the Science Theory, and see y'all next time.